So unfortunately, there's no C. elegans in this presentation. But it's a collaboration between, right down here, it's kind of small, the SSGCID, which is the Seattle Structural Genomics Center for Infectious Disease, DSTL and University of Würzburg. Both of those, DSTL is in the UK, and University of Würzburg is in Germany. And Emerald just renamed themselves Beryllium. One of the things working with a private company, they like to rename and restructure everything every few years. So we're part of a consortium that operates under a federal contract, similar to a grant, but we do structural biology for community requesters for different labs. And we also do our own work that we find interesting that we can fund through either the contract or some sort of external funding. So also I kind of want to plug the SSGCID to any labs here that might be working on things like T. cruzi or Leishmania or anthrax lethal factor or things like that. You can request protein structures with compounds or just ABO structures of any protein you might be interested in. And so today I'm going to talk about a protein called PPML1. And some of you might be familiar with FKBPs and the FK506 compound. And this is going to be somewhat involved in this. I have this nice little picture of the protein with one of the compounds that our chemistry collaborators have made, and it's found in the pocket that we're looking at. So, as many of you might have heard, there's a big need for antibiotics. There's some organisms that are hardly covered by current antibiotics, and a lot of antibiotics are expected to become ineffective in the next few years. And one of the emerging diseases is Burkholderia. It's the cause of melioidosis, and you see it a lot in Southeast Asia and I believe Australia. There's not that many cases in the United States, but NIAID, the National Institute for Allergy and Infectious Disease, classifies it as a Category B pathogen. It's also an organism that is a potential biological warfare agent. So part of the SSGCID is we work on a certain class of infectious disease organisms that are defined by the NIAID. And this one had quite a few community requesters and people interested in it that we could collaborate with. We don't do our own chemistry, and we have pretty limited biological assays. And so currently there's no vaccine and no good antibiotics for this target. So what's a good target in Burkholderia that we can use to develop antibiotics? But before we look at that, we have to ask, what makes a good therapeutic target? So we need to look at the target selectivity, whether this target is going to be found in other organisms that we don't want to hit. The same or similar structures that are found in humans, we don't want to develop a drug for something that's going to affect us just as much as it affects Burkholderia. Also, essentiality. We want to make sure that we're hitting a target that is really going to have a strong effect on either killing the organism or decreasing its infectivity. Another thing that we're going to be really concerned with is a proof of concept once we develop a couple ideas, and showing that it's actually feasible to develop a therapeutic and convince ourselves to invest more resources into a project. And then one of the great things about this target, PPML1, is nature knows best a lot of times. The compound FK506, which was originally used for immune suppression in humans, that is kind of an anomaly, and the compound is actually an antibiotic that was discovered from soil sample in Japan, I believe. And so all of this is kind of taking place right in this area in the whole scheme of developing a therapeutic. And this goes all the way through optimization and clinical trials and then regulatory approval. And it's pretty rare that any of these endeavors actually make it to drug approval. It's extremely hard to get to that point. It would take many years and a lot of money and some luck. So looking at BPML1, it's a peptidoprolyl cis-trans isomerase. And of the compound FK506, it's a macrolide. And we're specifically interested in this pipcholine anchor. And our chemistry collaborators developed a bunch of pipcholine derivatives. And the really important thing that we need to avoid is the wide range of human effects. This region right up here that binds to calcineurin and gives it the immunological effects that we see in humans. So a really important concern is avoiding that function in our drug discovery. And in Burkholderia, this is a MIPS, a macrophage infectivity potentiator. So it doesn't actually kill the organism. If we knock out this gene, it affects the infectivity. So then your own immune system can wipe it out when it becomes less effective. And another important thing is the novel class of antibiotics. Antibiotics only hit a few key targets, and it would really be advantageous to find another range of targets to hit with antibiotics. And also, you can see there's quite a few other organisms that have these FKBP MIP proteins. So we have a broad set of tools with our collaborators that can help us solidify this as a good target. And at DSTL, they're a defense organization that's part of the UK government. They have knockouts and Burkholderia assays, and they can do toxicity studies, and most importantly, the macrophage infectivity testing, so we can use compounds that we discover to see how it affects the infectivity. And at Emerald, we started out synthesizing quite a few genes, around 50 different constructs with different tags, to try and get the right protein for crystallographic studies. And if you guys aren't familiar with X-ray crystallography, we need very high amounts of very clean protein to set up crystal trials with. So it can be really difficult to make a crystal of these large 50, 60, 70 kilodalton proteins. And it turns out that this protein, BPML1, requires a His-Smith tag that is non-cleaved to be in the crystal structure. That's the only way we can get an apo structure without any compounds found. And some of our probe compounds, which are the pipcholine anchor molecules, are from the University of Würzburg. And then we also have NMR assignments that weren't actually collected by us. They were in the BMRD, which is a database of NMR assignments for residues. So we decided to use fragment-based lead discovery to try and find some new molecules that would bind to this same pocket where this pipcholine anchor is in this FKBP. And so fragments are usually small molecules between 100 to 300 Daltons. And at Emerald, we have our own library that we've designed that uses the rule of 3. It's usually compounds under 300 Daltons, a C-log P of less than 3, and hydrogen bond donor and acceptors usually 1 to 3 of those. And these are generally weak binders. They have pretty high ligand efficiency though, because they're small. The better that they bind, there's more of an opportunity to grow and expand those into more drug-like molecules. And there's a couple of theories behind using these small fragments of molecules. And one of them is fragment linking. We can get 2 or 3 of these into a pocket and then link them chemically and make a larger, tighter binder. And another option that's kind of illustrated down here with a compound that's actually an approved drug now is fragment optimization, or growing a fragment. And we have a bunch of different techniques that we can use to identify these fragments. And one of them is saturation transfer difference NMR, which we do a lot of at Emerald, and also SPR, ITC, and different NMR techniques, and also crystallography. You can set up a ton of crystals and soak compounds into each one and try and solve the structure of each one. So for this, we ended up using 2-dimensional NMR. We used a HSQC or HMQC ligand binding experiment. And so we made N15 labeled protein. So we're looking at the nitrogen proton bond on the backbone of the protein chain. And so up here we see FK506 binding to, and here's the whole spectra. And we choose these 3 reporter peaks that correspond to this valine, this glycine, and this, I forget what D is, spiritin. But we use these 3, and
Spartic
acid. And we can see over here, FK506 is a really tight binder. I think it's low nanomolar range. And we can see the slow exchange of the apo peak right here, disappearing and reappearing as we titrate in more of the compound. And that's because in the time frame of the NMR, you don't see the compound coming off and onto the protein. You only see it on the protein and off the protein. And then one of our probe compounds that was synthesized for us, we see a faster exchange and we see the peak shift. So we're seeing a range of states of the compound coming on and coming off the protein. And we use this to identify hits, and then also rank those hits on how much there's a shift in these peaks. Because each one of these peaks is correlated with the nitrogen backbone of the protein. And when something binds, it's perturbing the electronic environment of that bond. And so that's why we see the shift on the 2D spectra. So using these reporters, we did a fragment screen by a version of HMQC called SOFAST HMQC, which is band selective, optimized flipped angle, short transient HMQC. And if any of you are familiar with HSQC or HMQCs, they usually take a really, really long time to run these experiments. And this one only takes 10 minutes. So we could screen pools of compounds really quickly. And we have automated robotics to insert samples into the NMR. So we could screen thousands and thousands of compounds in just a few days. And then the bottleneck becomes somebody has to go through and look at all these spectra. I do a lot of that. And so our mixtures are usually 8 to 10 fragments. We can do more, but it gets quite a bit more difficult to deconvolute larger mixtures of these small fragments. And you're also more likely to have fragments competing with each other. And down in this little schematic, these fragments are floating around in the solution, and pretty much we're trying to identify which ones are hitting this pocket. And we do a titration so we can see a range of shifts so that we can rank them when we do find compounds that bind. And then also we deconvolute these by going back and titrating in one compound at a time till we see the same shift that we saw in the mixture. And so from a screen of 1,500 compounds, we saw these 4 hits as the best binders. They had the greatest shifts in our reporter peaks. And 3 of these are pretty similar, which I thought was really interesting. We have thousands of compounds of all sorts of different things, and we see these 3 same. It's one moiety. And there's this other compound that did pretty well that's relatively similar in shape to our picoic anchor. So this moiety that we see is also found in a lot of other approved drugs, which gives us a lot more confidence that we could develop these as a therapeutic, and they might be interesting leads. So here's the spectra for one of the compounds, and we see the shifts in these 2, not quite as much this one. And hopefully the reasoning for these large shifts will become apparent if we get a crystal structure and we can understand the mechanism of binding. We also see some bigger shifts in other peaks. These might be nitrogen side chain peaks. And we see some other perturbations down here, which are all indicative of pretty good binding for a fragment this size. Then with our other compound, we see shifts, larger shifts in this one, but smaller shifts in these 2, implying that it's affecting these residues differently, which makes sense. Those were 2 different moieties. So we really want to chase these up and see what the next step is.
Yeah, I think I missed something that you might have said. What are the axes on this plot here?
Oh, so this is going to be your nitrogen, and this is going to be your proton. So we're looking at the nitrogen-proton bond on the peptide backbone. Did that answer your question?
A little bit, yeah.
So in an NMR spectra, this is the ppm, the range of, so like in a normal 1D spectra, you'd have a spike here, or peaks, that would correlate with a proton. And on the nitrogen, you would have the same thing. But in 2-dimensional NMR, we get these 2-dimensional peaks that end up being kind of like topographical points on the spectrum.
So there are places where there's a peak on both of them. You then get that dot.
Yeah, so it's the bond between the proton and the nitrogen in the HMQC.
A separate question. So those 3 amino acids that you're looking at, are you looking at those particularly because you know that's the normal FK506 binding site?
Yeah. So
because there's all kinds of shifts in other areas, and you're not really focused on those.
Yeah, correct.
Okay.
Those other ones could be just as important as these, but we need to make a standardized way to compare all these thousands of molecules. And we picked these 3 because if the compound is right here, we have these 3 points that make kind of a triangle around it.
Okay.
So if it's binding anywhere in that pocket, it's probably going to perturb at least one of those.
Okay.
And so then we did some StructureActivity Catalogue, StructureActivity Relationship experiments by buying a bunch of different analogs to see if, without crystallographic data, if we could expand these hits and maybe see if we could find better binders. Just looking at random analogs and making guesses and seeing what was commercially available. And then doing pretty much the same experiment with those hits, and we didn't find anything that made a big significant difference, which kind of highlights the importance of having the structural data to guide us. Because without it, we know these are hits, but we don't know how to make them better. We don't know how to make them bigger. And that's what we're ultimately trying to do. So we don't even know which direction we need to grow these. If we want to go this way or change things over here, we don't really know what's important for binding and where we want to go to make it better. And also at the same time, we have the second approach where our medicinal chemist friends are making these derivatives of the picolictic anchor that we know. We know the orientation that it binds because we have the crystal structure of FK506 and this BPML1 protein. So with these compounds, they're already getting much bigger. We have some pretty good binders, the low micromolar range, and we have crystal structures of all these. These correspond with PV entries. The protein database is where we upload all of our 3D crystal structures. And so we can also use these as probe compounds to test our initial hits and see if we can add those in and they'll outcompete our fragments. And a lot of them do. So doing structure-activity relationships on these, where we have crystal structures, we've identified these 3 regions that are potential areas to grow these compounds. And also our reporter protein, or our reporter nitrogen signals are going to be right up here, right over here, and right down here. So growing in any one of these regions, we would expect to see a shift in these 2 if we're growing this region, or a shift in these 2 if we're growing this region, and the same with this region down here. And what we ended up finding was this Q area is not a very good area to try and grow our molecules. There's not a lot for compounds to bind with down here. We did find that this area could really benefit. Here's a different view of it. Could really benefit from having a longer linker on this XY region up here. And maybe we can make some hydrogen bonds up here and make this a tighter binder and a better molecule. So we've done in silico studies of that, and we're doing some iterative rounds of chemistry to get some different compounds. And comparing that with what hopefully we'll be able to get soon is some crystal structures of our hits by NMR. And possibly use that to either combine those compounds or use one of them to guide the growth of our NMR-derived compounds. And also we've got a NATO grant for this because funding ended for this project. And I didn't even know NATO funded science projects like this until we won this grant. But apparently they're interested in potential biological warfare agents. And so in addition to trying to get the crystal structures of these compounds that we've identified, we're continuing to screen more libraries and more compounds to find hopefully a larger set of potential starting points for therapies. And then also some future work is comparing this to the effects that FK506 has, because we really want to stay away from the complex that sequesters calcineurin to avoid the human interactions that reduce the immune system in organ transplant patients. Because FK506 is used to, for when you get an organ transplant, there's the host response, and FK506 knocks down your own immune response to it. So we want to avoid those issues. And a lot of people have worked on this. Quite a few people. Darren Bagley is my boss. I thank him. And all these people at the other collaborating places in the UK and Germany and at Seattle Biomed and University of Washington.